Welcome everyone, and good morning on this side of the pond, and good afternoon to Professor Paolo Coppi, and good morning to Professor David Hackam from Hopkins. This is the second webinar on basic science that we are doing now for UPSA. We've been running a series of webinars with more or less once a month on mainly clinical topics, and now we dive into basic science. So we had a very successful one on CDH, and I'm sure this one will be absolutely amazing. Today we're going to talk about bioengineering of the gut, and we have two giants that are joining us. My co-host, Martin Lacher, is going to join us soon. So, but without any further ado, I'm going to introduce Paolo Coppi, who doesn't really need much of an introduction. Is a friend and a colleague. And for us in UPSA, he's actually just been elected as the incoming president. So that, I would say, is the biggest thing on his bio present for us. Paolo is the Nuffield Chair of Pediatric Surgery and NIHR Professor of Pediatric Surgery and Consultant Pediatric Surgeon at Great Ormond Street Hospital in London, UK. Is the head of the Surgery Unit, Stem Cell and Regenerative Medicine Section, Developmental Biology and Cancer Program at UCL, Great Ormond Street Institute of Child Health. Paolo comes from Italy, like myself, from Padua. He got his medical degree in Padua University, but he spent time in Boston working with Anthony Atala and publishing a seminal paper on the identification of amniotic fluid stem cells. So that was published on Nature Biotechnology, but even more, I would say, on Metro magazine. I remember that day, and it was very well publicized worldwide. Paolo has has supervised more than 35 research fellows and PhD students, and he has been awarded various national and international grants in excess of 30 million pounds, British pounds. So you can do your maths and convert that to euros. So his main interest is tissue engineering of the esophagus. And so, Paolo, over to you. Tell us what you're doing in your lab at the moment, and what the challenges have been, and tell us all about your cutting edge research.
Thank you very much, Augusto. Thank you, Professor Zani and Professor Lacher, for the kind invitation to this webinar that are becoming more and more popular also among residents and registrars. So I think this will be an opportunity, hopefully, to inspire some more registered to do research in this field. And I think Professor Lacher is connecting, which is great, perfect timing. And it gives me a great pleasure to share this session with Professor Hackam, which I always admire his work, and he's certainly one of the pioneers in the field. So I will start sharing my presentation. Hopefully you will see it now. And I will focus during my presentation certainly on the role of the extracellular matrix, because that's something that is very close to what we do in the lab. I need to declare some conflict of interest on the amniotic fluid stem cells and also on gels created from the extracellular matrix. So where I'm talking to you today is actually from this new institute for research in rare disease in children, which has just been opening during the pandemic here at Great Ormond Street Hospital. Is dedicated really to the discoveries that hopefully one day will be translated on the opposite side of the street to the hospital. And we are very fortunate to have this institute here, which I know over in the United States and North America looks at small building, but over in Europe is certainly something that we go very proud of. So when we discuss about approaches for engineering, we have learned a lot from something that happened about 11 years ago. Here is Kieran that was born with a congenital trachea stenosis. And at that time, after several operations, he reached the point where a stent was eroding through the trachea into the aorta. And at that time, something different needed to be done. So Martin Elliott that led the cardiothoracic team. We took a deserialized trachea from a cadaver, and we utilized that with cells derived from Kieran to transplant Kieran. Kieran is now 22 years old and is doing well, is an adult, so he's not anymore a gosh, but it does just follow up with the adult, but is doing extremely well. So we learn a lot about how to use matrices to repair tissue or organs, but we needed to do much more if we wanted to do something more complex. And one of the issues that we really come across with the engineering is the fact of vascularization. So when you have a tissue or organ, you need to vascularize that. And the trachea at that time, as a general surgeon, my only input as a surgeon was to wrap the omentum around the trachea. But if you want to irrigate a gut, for example, this is certainly not enough. And that's why we were very fortunate to work together with Shaheen Rafi over Cornell University, that is an expert in the field of endothelial cell biology. And the idea of Shaheen was that why we don't reprogram these cells back to more an embryonic stage. And to make a long story short, using ETV2 as a transcription factor, which is not expressed during the adult life, we reprogram the cells back to a benzodone precursors. And by doing that, we noticed that the cells were able to grow much rapidly, to form capillary for much longer in vitro, and also to create vascular network. So those vascular network could be perfused, and we could use bioreactor to really demonstrate these, like you see here, with particles going from one compartment to the lower compartment through this network. But then the clue experiments was to take these endothelial cells and put in a customized bioreactor that we build in the lab, where the cells can go from one compartment to the other, but using human blood. This is human blood running through the endothelial network, and you see there's no leakage. Here the endothelium is in green fluorescence protein, so GFP, and you see the human blood running through that without forming clotting and without extravasation. So then we thought, well, this is maybe important for engineering. And so in our customized bioreactor, we try to use that to recellularize the vascular endothelium of an intestine. So you see here an intestine with this mesentery, and the intestine that gets repopulated by this endothelium. And what we observe is that the endothelium was able to colonize all the peripheral vessels. So this overcome one of the main problem on regenerative medicine in general and tissue engineering is about the revascularization. So you see here how the endothelium of the mesentery is completely covered by these ETV2 cells that get reprogrammed. And this is valid also when you only conditionally reprogram. So the cells are not always expressing ETV2, but they're expressing ETV2 during the repopulation of the organ, and then get switched off. This results doesn't change. So can we jump then to repopulate the old gut? Well, that's very difficult. I'm very fortunate to be part of a consortium that works in Europe mainly, but also across the Atlantic, on building a small intestine or trying to build a small intestine for children that have a short bowel. And we have learned a lot in the last few years working together, but we learned that it's also very difficult to do so. So we have been working on, for a number of years now, trying to deserialize intestine, and we can create intestine that deserialize but contain still all the structure which is normally present in the intestine. You can do that also with human, and for who is interested, we're very happy to share protocols and so on to do this in any of the pediatric surgery department that are connected today. We can deserialize efficiently also human colon. And you see the structure remain. So here is a colon that you see the crypt. You don't see the villi, of course, in the colon, but you see the crypt maintained. And we can also to understand the structure of the colon. So in Grenoble, there's an accelerator, and we've done some experiments to understand a bit more the three-dimensional structure of the intestine. And using this type of modality, we can then use mathematical modeling to try restructure the intestine. And actually here is some of the first few attempts that we've done about using the cell small intestine in this case as a model to print out a new small intestine. So you see here villi and crypt present in the printed. Of course, we are surgeons, so for us it's pretty much the same, but actually scientists will tell you that there's quite a bit of difference there. So which material to use to print? Well, this should be a material that allow the organoids, which are really the stem cell of the intestine, to grow and to proliferate and eventually to differentiate. So we say, why don't we use the natural material? So the intestine, and here is just some intestine, it gets deserialized in vitro. We understand much more about the ECM now. So we, for example, see that there are clustering of ECM. So you can notice here in the C figure that there are cluster of the protein expression of the ECM, which is different. I mean, pretty much recapitulate what are the three germs layer, but we identify at least 4 cluster in which we have gut, liver, and lung, for example, endodermal organ clustering together, and some that are ectoderm clustered together as well. So we know that the ECM do not only contain information that are mechanical, but probably also the biochemistry of this ECM needs to be investigated further. So we are being able to culture organoids derived from the stomach, from the liver, or from the small intestine quite reliably in this way. And actually, I want to highlight something that hopefully is coming out soon, which has been led by Giovanni Giobbe in the lab, which has been using this type of technology to actually study COVID. So we saw initial, and I'm sure many of you have observed that in children, we had these GI symptoms in children with COVID. And in fact, we are now able to see that there's some subtle differences between pediatric gastric organoids and adult gastric organoids that could actually explain why maybe pediatric is more susceptible to GI infection and COVID. And but the real aim is actually to grow these organoids to make a mucosa. And indeed, we focus now on the small intestine mucosa because that's what is really lost when you have a short bowel, like the volvulus or in gastroschisis. And work that we have done with Vivian Lee for the past 5 years has really focused on trying to grow organoids derived from children with short bowel syndrome to see if we could expand those organoids and maybe one day, for example, substitute the mucosa of the colon. So we don't need to rebuild the old intestine, but we could use the colon, which is still there, to have a mucosa that is absorbing and secreting. So the first trial of experiments that we did was to prove that we could expand separately duodenum, jejunum, and ileal organoids and expand those while maintaining their signature. And indeed, that was the case. So we then focus on the jejunum organoids, and we could specialize them to some of the characteristic specific to jejunum organoids. So we could expand and actually differentiate them. And so the final bit that we wanted to do is, could we see them in D cell colon and D cell intestine and show there's no difference? And indeed, that's what we've done. So this was human small intestine and human colon that was deserialized, and seeded then with jejunal organoids. So first of all, the D cell, small intestine, and colon do not differ much. So in terms of protein, there are very little differences there. Of course, we know the structure is quite different, but the composition of the matrix is not very different, and that's important. So we seeded this in, again, customized bioreactors, and we were able to actually see that the cells were reaching some degree of specialization, and who is interested, you can see more in the published work. But most importantly, we were seeing in the colon seeded some rudimental villi with newly deposited matrix from the organoids that were seeded. So this is quite interesting, because it means that hopefully in the future, we will be able to transplant a small intestinal mucosa in the colon. So are we ready now to jump to more engineered organs? Well, as Augusto was kindly mentioned at the beginning, we are very much focused on one part of the gut, which is upstream, which is the esophagus. And I'm sure I forgot some of the people involved in the work, but certainly these are some of the key people that have been really involved in the work that is trying to culture esophageal cells outside the body and to seed them in deserialized esophagus. And this work is in collaboration with Paolo Bonfanti and at the Crick Institute and Giulio Cosso at the University of Manchester. So we have shown now for a few years that we can deserialize the esophagus, and we use bioreactor to reserialize efficiently. We have seen over the years that some key elements are very important. So you need to co-seed the fibroblast with the muscle cells. These allow the muscle cells to migrate in the matrix and actually to differentiate. And this is very important. And I'm sure Professor Hackam would agree that actually fibroblasts play a key role in many of the components for regeneration. We use then bioreactor to somehow reprogram the cells to know what they need to do. So like contraction, and we can follow them up using luciferase. And we can also see that the bioreactor make the esophagus much stronger and thicker. I'm sure many surgeons would be happier to suture the esophagus on the right than the one on the left that hasn't been conditioned in the bioreactor. So Paolo, our group has focused their work also on isolating the epithelium and reliably culturing that, and then using a preconditioner of the esophagus in the omentum, we can seed in the second stage the epithelium to seed it. And in fact, the muscle can differentiate quite efficiently. And what we have seen, and this is the last couple of experiments I'm going to show you, is that we can implant now in animals. We've started with rabbit and we are now in the pig. And what we can see is that we need to use a stent on the esophagus at the beginning. So you see here is a rabbit. So we know that the vascular supply is important. Here you see a patch of muscle wrapping the esophagus. And then you will see a section that demonstrates how you need to have a stent that helps the initial part of the structure of the esophagus. So we see here a CT scan just cutting through, and you will see the stent appearing. These are PDS, essentially stent, are called Ella stent. We use it clinically, and are absorbable stent, so we can leave it there without cutting. So I think it's important to recognize the role of stem cells, which in the last 5 to 10 years have really changed. We know almost as much as the stem cell of the intestine as the bone marrow nowadays, which is remarkable. There are still unsolved problems, so we are not ready for prime time, but certainly regenerative medicine is a clinical reality. So I think many of the surgeons will be more and more involved in this aspect. So finally, a picture without mask. I couldn't believe it when we took it. We were allowed outside to take it. And I could spend a long time thanking each and every one of the member of the lab. Of course, we eat only Italian, so that's why pizza, and all the funding body. I would like to thank also my clinical colleague, and thank you very much for the opportunity.
Thank you, Paolo. That was, as usual, an amazing talk with a lot of new data also that you were very generous to share with us. So it's really interesting. I see that Martin has joined us, and just before I leave you the microphone to introduce Professor Hackam, I wanted to say that we've got a good number of participants, but we are streaming actually also on Facebook and on YouTube.
Good afternoon, everybody. Thank you, Paolo, for the excellent talk, and it's a pleasure for me also to be here. So let me introduce the second speaker of today before we start the discussion. It's Professor David Hackam. He is Surgeon-in-Chief at Johns Hopkins Children's Center, obtained his bachelor and medical degrees at the University of Western Ontario, then earned a doctorate in cell biology from the University of Toronto, where he also did his general surgery training, and then he completed a surgery fellowship, pediatric surgery fellowship in Pittsburgh in 2002. Then he joined the faculty in Pittsburgh until 2014, when he became the Garrett Family Professor of Pediatric Surgery at Hopkins University, where he is the Surgeon-in-Chief. So Dr. Hackam's lab is focused on unraveling the molecular mechanism that underline a lot of important diseases like NEC, IBD, or trauma. And he seeks out actually, and that's, I think, is special, to develop novel therapeutic strategy to prevent and reverse those processes. And it was actually his group, and a lot of people are aware of that, who discovered that NEC is caused by increased expression of signaling of TLR4 in the newborn intestine. So David, before I tell your entire story, I just want to, which includes rodent models, intestinal stem cell isolation and culture, tissue engineering, and parents, and you have got funding through the roof. But I think it needs to be amplified that they are still the surgeon and a great teacher. So one of your focus is also to be very passionate about training the next and the current generation of clinician scientists at all level of training. And so that's why it's a big honor for UPSA and all of us, David, that we could get you on board for this session today, and for the session Cool Kids to Basic Science. You're one of those cool kids, and the stage is all yours.
Thank you very much. Those are very kind words. I'll never be as cool as someone like you guys with your cool accents and all that great people you have. Just pay no attention. So this is my screen. Thank you for the invitation. I'm glad I could move things around to be here. Zoom makes everything both easier and harder. A couple of disclosures. First, I got my third shot of Pfizer vaccine yesterday. So great. I think it's great, except I think my TNF alpha and IL-6 levels are through the roof. And so I feel like I got hit by a lorry or maybe even a meteor, but it is what it is. And I encourage everyone to get vaccinated. It's a real pleasure following Dr. Coppi, Professor Coppi. Paolo, I've followed his work for so many years. He's truly a leader in the field, and did me a great honor when I was a leader of one of the American Surgical Societies to be a featured speaker, and people are still talking about your talk as sort of the high watermark for that meeting. So it's great for pediatric surgery, great for surgeon science. But I was asked to speak about tissue engineering, so I will be focusing these remarks on tissue engineering, which I think will be complementary to some of the real groundbreaking work that Paolo presented. Martin, Professor Lacher talked about my passion for training, and I know we all share that on this call, certainly. But I want to open just with this slide. These are the faces of the people that did a lot of this work. These are the surgical residents in my lab that performed a lot of the studies that I'll be describing. And I always put your name, your choice, because I just gave this talk to the Department of Surgery, who are always interested in looking for opportunities for research. But these are not the postdocs, the graduate students, or the MD, PhD students who are wonderful and incredibly important. These are our brothers and sisters, general surgery trainees. And I think, as Paolo and others would agree, surgeons are well suited to do science as long as the environment is supportive and the question is relevant and the environment is structured. I said I wouldn't talk about NEC.
Sorry, David, I'm deeply sorry to interrupt. Could you go on full screen mode, because this would like enlarge your presentation?
Yeah, and
much nicer.
There's no problem interrupting me. I'm going to try it. Sometimes it works better than others, but let's see. All right, can you see that?
It's much better now.
Perfect.
Okay.
Yes.
Good. Thank you for that. I said I wouldn't talk about necrotizing enterocolitis, and I won't, except for the parts of the talk where I do, which are just at the very beginning. And so tissue engineering and our focus will be on the small intestine, needs to solve a problem, and the problem is massive intestinal loss as occurs in kids that have sustained NEC, but also volvulus and other intestinal loss. We do hope to prevent this disease and to treat it. But the beginning, I need to just share a couple of neck slides before we launch into the solution, which is tissue engineering, because I want to put that in the appropriate context. It's always important to disclose that I do work with companies, especially since neck is a disease that is largely seen in the setting of infant formula administration, the absence of breast milk. So I work with formula companies to develop formulas that are neck protective, as well as other companies that we hope will bring our discoveries to the clinic. I also have to acknowledge this book, and thank you to many that contributed chapters, and also those that have purchased it. It's on Amazon. You can search on Amazon. It's not really a plug for the book. It's mainly a way of saying thank you, and that there's a one-stop resource to go to for your trainees, for yourselves, for this book. And this is the pathogenesis right here of TLR4 in the intestine and the dysbiotic microenvironment that we know these preemies have when they have formula. And it activates this signaling cascade that causes inflammation and intestinal distension and neck. It is a bestseller. It's 1,248,610 on the Amazon bestseller list, where I know you share with me your excitement for the movie to come out. And now that the last installment of James Bond is out, I think that actor has time on his hands, and I'm just waiting for him to call to the screenplay. Very briefly, what happens in our lab as we look at neck before we talk in a little more detail about tissue engineering, and I'm going to keep this to about 20 minutes so we have time for discussion, is like all of you, these are the questions that we think are important. We're interested in what causes neck, and as a corollary, why are preemies at particular risk for this disease, and is there some aspect of development that we can enhance? And the answer is yes. And can we prevent it? And since breast milk prevents it, can we learn from breast milk how breast milk works? And the answer seems to be yes. Can we predict it? The answer to this appears to be maybe. And we're integrating the the electronic health record and machine learning to come up with predictive models. And then the focus of the complications of neck. Babies with neck have neurocognitive impairment, as most beautifully recently shown by Marty Blakely's group, significant lung injury, and for the purpose of this conversation, intestinal loss. And just one overview slide, and this is what's happening in the lab. You're very kind to mention our work on TLR4 dependent pathways. We think that many clues in the pathogenesis of neck will be found by looking at how TLR4 signals in response to the abnormal microbiome, and how probiotics and breast milk can influence the microbiome and shut down this developmental program. And just so people are aware, NEC occurs in the presence of elevated TLR4 because TLR4 is a developmental gene. It's required for gut development in humans, in mice, in every species we've looked at, piglets, monkey, and the fetus was supposed to be born in a sterile environment, of course, and so TLR4 levels are very, very high. It turns on Notch, it turns on Wnt, all these important genes for development of the gut. And so when the baby comes out prematurely, these developmental genes are still turned on. But now the sterile womb is not so sterile. It's a dirty NICU, no offense to the NICU. But now these developmental genes see bacteria. Unfortunately, they're also bacterial recognition genes. Don't ask me why. I didn't design it that way. But these developmental genes have bacterial recognition roles causing inflammation and NEC. And so there are clues that link development and disease pathogenesis. We're interested in the effect of diet and metabolism on NEC, especially certain ligands. Prediction looks at maternal factors, machine learning, and then the long-term complications. And we're going to focus not so much on the brain or on the lung for the purpose of this, but on the artificial intestine. And just one recent story, because it's 5 days old, and so I guess 7 days old. Time flies. I point you to this paper that just came out. The first author, Mark Kovler, is a surgical resident. He's going on to become a pediatric surgeon. And where we showed that this bacterial receptor, one of the things that it happens to do is it cuts down some of the enteric nerves in the gut, and especially the enteric glia, which are support structures that nourish the nerves. And their loss is critical for NEC. And this finding was based on the observation that we all make, that is, every baby with NEC first has abdominal distension and feeding intolerance, and essentially an ileus. And we all believe that this ileus is a consequence of NEC. And so we thought, since it occurs so early on, and since Bell staging, Bell 1, Bell 2A, is abdominal distension, feeding intolerance, maybe, which is a marker of an ileus, perhaps, maybe ileus is a cause of NEC, not a consequence. And in fact, that's what we showed. We showed that ileus occurs because TLR4 knocks out the signaling molecules that allow the cells to work. Breast milk actually restores these, and we identified a drug, which we called J11, which turns on these glia once again and prevents NEC, as you can see here, and it works in human tissue ex vivo very nicely. So that's just a little bit of a NEC story, and this is a sort of what happens when those approaches fail. What happens when NEC develops and as surgeons we operate and we resect the intestine, and now the baby's got short gut. And the work of Paolo and others across the field and other fields are working on this question of developing an artificial intestine, which is the word we use, and the rationale should be apparent. And this is how, that was just like a subtle Freudian slip. I did not mean to show it twice, but it's like advertising. If you just flash, buy Coca-Cola and you don't realize, or sorry, eat pizza and you don't realize that at the end of the talk, you'll be hungry and you'll order a pizza. So if I just flash that book, I'm just kidding. I didn't mean to do that. So these are the components of the artificial, and I receive no royalties, obviously. It's just for the field. I just wanted to say that. So the components are cells, scaffold, and the environment. And we talked a little bit about some of these in the previous talk. Dr. Paolo has really done a lot of important work in this. And I'm going to sort of touch on the stem cells, a little bit on the scaffold, and not a whole lot on the niche, which Paolo described. This is our workflow. And we do this either in mice or in piglets or in dogs, where we start with the isolation of the stem cells and we put them on an appropriate scaffold. And the scaffold is critical, as you heard about. Blood supply is essential, and we do use the omentum. And you'll see complementary work from our findings, which really support a lot of Paolo's work. And then we feel that we can bridge the ends and have a blood supply to nourish and absorb. The stem cells we're talking about are not embryonic stem cells. We do not use embryonic stem cells. We use these stomatic stem cells that will give rise to the epithelium. And the more undifferentiated they are, they will give rise to all of the epithelial progeny, including enteroendocrine and goblet and Paneth. And the Paneth cells are so critical for supporting these stem cells. And these are the progeny. And when working in human tissue, which we have to do, we work with induced pluripotent stem cells. These are fibroblast-like cells, which we de-differentiate into intestinal stem cells, and then mature intestine, which led to a Nobel Prize, and that's the workflow. And so just a couple of slides to show what these structures look like. This is from the somatic stem cells. This is not IPS. You get these beautiful structures with each of the different progeny. You can do it from mouse and human tissue. Just some videos here. You can see that down here, they do form these crypts, and they do shed into what is essentially a lumen. You can see that right there. And there has been interesting, this is worked on my surgical resident, Dr. Laura Martin, who's now a pediatric surgeon in New York. And you can see the three-dimensional structures. This crypt is coming out at us, and they fuse together. They, under different conditions, they will form cysts where they secrete the material into the cysts. They'll differentiate and branch, but only so much. And we're very interested in how that happens. And we can modify this with various bacteria. That's a probiotic. It's impaired in enteroids in the presence of NEC. And so the scaffolds that we use are engineered. They're not 3D printed, but they are laser printed into a biomatrix. And this is work from our collaborators at Cornell University, where we get these delicate 3D structures that look very much like the native intestine. And you can see we can tubularize this, and we get these villi, and they're absorptive, and all they need is a blood supply. And they cover very nicely with stem cells. And again, this is the workflow. And so we need animal models to test this. And so I'm going to take you on a little bit of a zoological tour, starting in mice, where this is the type of structure you get in the omentum, where just a cross section through some of these villi structures. And you can see that over time, we do indeed get a blood supply, which is shown here. We get the endothelial cell progenitors are attracted to the area, and you get this fine network. This is von Willebrand factor. And that absolutely requires the enteroids. The scaffold alone doesn't seem to do it. And it's not as if the mouse itself is putting stem cells in. This is all donor origin, which is very important because of the potential clinical application for these. And this is just evidence that it's donor cells and that we recruit immune cells. And you can see in a very, over time, in a bioreactor, you can get all these various tissues. You can get the epithelial cells, you can get the immune cells, et cetera. And I guess science thought that shows promise. So that's the mouse work. It's an epithelial stem cell progenitor pool that grows on a 3D scaffold that gets a blood supply and works in a mouse. So there are obviously questions about the enteric nervous system, peristalsis, degree of absorption, scalability that are still open questions. But we are not mice. Maybe we're a little closer to dogs. So we've used the same approach. Basically, we do a Suave procedure. You can see this is the Lone Star that we use in our kids, not the exact same one, at least I hope it's not the exact same one. Now I'm a little bit worried about it, but because it looks pretty similar. But we wash it, or at least we rinse it under hot water for, I hope we rinse it. Anyway, so you can see we're starting a Suave, and we do a mucosectomy, and then we implant from the mucosectomy our graft in here. And the advantage of doing this in a dog is that we can then perform proctoscopy, and you can see the suture line here and the graft proximal, and how it looks like a very nice mucosa. What this gives us over the mouse is this is where this graft is supposed to be. It has an intact blood supply, where we are implanting a scaffold. In this case, it's absorbable. It increases the surface area, prevents stricture formation. If we just put the enteroids in there, it just scars down. And so that's why you need a scaffold. And you get a little bit of a sense that the scaffold gives you goblet cells there. We're perhaps a little closer in infants to piglets. And so using a piglet approach, this is a piglet that has had our artificial intestine, has a central line, and these are the piglet's friends. This piglet is supposed to be in the cage, so I'm not sure how the piglet escaped. The work in the piglets is shown here essentially, and I'll just draw your attention down here where you can see we're implanting the scaffold with the enteroids, and we leave it in the bioreactor from anywhere between 2 and 7 days. And as Paolo said, the ability to sew this is absolutely critical for surgeons. And so the design of the scaffolds, of course, has to have a 3D surface that allows incorporation, an adequate surface area, and vascular ingrowth. That's all fine as far as it goes. But if we can't sew it because it melts away or won't hold a stitch, it's of no use to us as surgeons. And so it's actually a real biomechanical engineering challenge, which we work on with our biomechanical engineering colleagues. And you can see we have good success over time. I'm just going to show you, and we bring out stomas, and we've implanted this upstream of the stoma, downstream of the stoma to assess the role of stool passage in enhancing or not enhancing. And in situ, this is a piglet that we've done a redo laparotomy. You can actually see, this is laser angiography, that our graft is up here. In this particular case, it's not in continuity. And you can see it's got a beautiful blood supply. And so this is a segment of a couple of centimeters, you can see there, of pig enteroids on scaffold implanted with a mesentery around it. It's in discontinuity from the bowel. So even though it looks like the kid has, the pig has an obstruction, the pig doesn't have an obstruction. And then we could put that into continuity and absorb food. And this is the 3D structure. And this is what this looks like at autopsy, and the kind of structures and the epithelial coverage that you can see. Before and after implantation, we want some degradation, but not too much degradation. And this is an area of ongoing concern. We need the scaffold for long enough to preserve the 3D structure, to have an influx of myofibroblasts and other fibroblasts that are going to maintain the structure. It has to have elastic properties. And so this interface between scaffold and enteroid and blood supply is really, in my view, the key unsolved or incompletely solved area. And you can see we get beautiful attachments and pockets where blood vessels have grown in. The porosity is one simple marker, and you can see these mini structures that we get. Dr. Coppi beautifully illustrated the work that he has published on a decellularized intestine. I think this has great merit. We performed similar studies, and it's very straightforward, actually, to decellularize and then to recellularize, which you can see right here. And I think this actually has great merit, and we've implanted this into pigs and shown great structures that seem useful. But in the final minute or two, I want to turn back to the human application. And so if you have a child who's lost intestine and you bring their intestine back together after resection, the residual bowel will adapt. And that adaptation is very instructive to us. If we can uncover the signaling molecules that drive adaptation, we can achieve two things. One, we can simply enhance adaptation in kids with short gut by identifying pathways and augmenting them or inhibiting them using new drugs or gene therapies. Secondly, if our goal and Paolo's goal is to develop an artificial intestine, wouldn't it be great if the matrix liberated certain molecules that actually enhance the adaptation of the epithelium on top of it? And I'm just going to give you, so this is, again, the type of refocusing that we need to bear in mind that others have talked about. This work is moving forward in multiple labs, and we're certainly learning from each other, and I'm learning from Paolo and others, Tracy Grickscheit and Mike Helmrath and Jim Wells, and lots of labs really around the world that are doing this type of work, and many, many others. But there are key barriers, at least in the United States, the FDA, there are so many steps that may pose risk, not so much the intestinal cells, which we think would be patient-derived or a decellularized matrix, but, and in fact, the same patient-derived. So we would take their skin fibroblasts and dedifferentiate them and then redifferentiate them. But the steps of purification require FDA compliance. And so we try and model after industry standards whenever possible. There are ethical concerns. It's not cheap. And of course, you got to get the right patient. I always return to the beginning. These are the surgeons that did this work currently, who are working in the lab and sewing intestine together and culturing stem cells and decellularizing matrix and feeding mice with neck and just having a good time. Summary is here. The proof of concept, I think we've established. Others have too, for the development of an artificial intestine. There's biocompatibility. Absorptive studies are promising, and the work showing the transcriptomic analysis of novel genes to reverse short bowel. This is a broader view of the people that have done the work over many years and the funding that has allowed it to happen. And in happier times before COVID, this is the type of picture we can take. Again, I'm truly honored for the opportunity to be part of this symposium, and thank you for the opportunity to present our work.
Well, thank you, Professor Hackam, for this excellent talk, and also Professor Coppi. Yeah, I think we are ready for discussion. I will look in the chat. Maybe Augusto looks at the chat and maybe ask the first question. So Paolo, you mentioned that you can sort of substitute the colon by replacing it with a different mucosa, like a small bowel mucosa. Does it matter where the colon lives? So could you actually take a segment of colon, interpose it, relocate it between two loops of small bowel? Would this like epithelialize differently because the microbiome is then a small bowel microbiome? Would that make a difference, or does it not matter?
Yeah, thank you, Martin. I mean, first of all, let me remark, I really enjoy, as always, the lecture of Professor Hackam, and it's very complementary to what we are trying to do in the lab. Regarding your question, you make a very good point. I mean, people have shown that you can transplant organoids from small bowel into the large bowel, at least in mice, and these organoids in a damaged colon can actually repopulate. I mean, Hans Clevers has shown that Toshi Sato is leading really this work, and was published in Nature last year, in which he was able to repopulate organoids in the large bowel. And you can think about doing that, for example, for diseases of the adulthood in the colon. IBD is one of those. But actually, if you think it's possible that the mucosa could be substituted to an absorber one, like the jejunum or allium. But you're making a very good point that actually the microbiome is still a problem in the colon if you don't interpose that to the small bowel. So it's possible, as you mentioned, that actually if you transpose the colon and then you do a you take away the mucosa,
Yeah.
sorry, of the colon, a mucosectomy, you can then substitute that with a small intestinal one that you have engineered in the lab, and then you transplant that into the colon that you have interposed. Mm-hmm. But it's quite interesting that, I mean, it's not very well documented, but there are some reports showing that actually in some children with short bowel, part of the mucosa of the colon become small bowel. I mean, there are biopsies that demonstrate that. So you can sort of promote that regeneration process, a bit like Davis was mentioning before.
Mm-hmm. Thank you.
Let me echo what everyone said. I mean, Dr. Hackam, you have given us an amazing lecture, and especially very generous to share some new data with us. It's incredible. You are producing so much on the front of neck, and here you are giving us a half an hour lecture on something that is close but not that close. So it's phenomenal. If we go to our chat, Francesco Lacconi from France is asking something that I think you both alluded to, which is the nerve system. Of course, we've all seen that everyone, including Mike Helmreich and Tracy Grigscheit, everyone is starting, of course, with the epithelium. But we know very well that there's other cell populations and, of course, the nervous system too. And how far are we from having really a full package of cell populations, tissue layers that would represent the intestine? And then the other question still from the same person is about the tubularization procedure. How do you test the strength of the scaffold? This is for both of you.
Okay, I guess I'll go first. Thank you for the kind words, and means a lot to me. Thank you very much, and say hello to everyone in Toronto and at Sick Kids, my old stomping grounds. That's where I did my PhD and had my initial training with that incredible group there. So the first question I think is very important. The role of the enteric nervous system. You know, and I'm glad people are asking the question, right? It's like, it's pretty amazing that the cells, the epithelial cells self-organize and that they cover. And then it's pretty amazing that they absorb. And then it's amazing to get a blood supply. And yet the field wants, but wait, what about, you know, it's like my kids. I can never like make them happy. So I like when they're asking for more, because it, you know, at least they're asking and they're engaged. So that's good. If you read Tracy's work, it does look, but the barriers are that it's not just getting primordial nerve cells, and we know the ontogeny of our enteric nerves very well in mice, less so in humans. There are reporter strains, and we've generated knockouts, and we know the subtypes of glia that are required for the subtypes of enteric neurons. This is important work. That we can adapt. So I think now the foundations are there. The trick is being able to grow these cells at a stage where they can differentiate into mature neurons and glia, and that they can connect. And then it's not just the ENS, but it's the muscle. If they have nothing to hook up to, coordination of peristalsis is very difficult. And so maybe you don't need nerves unless they have some immune or hormonal role, as glia do. If you can have a matrix that has some endogenous peristaltic activity, or if you can have an exogenous pump-like system, maybe the role of nerves can be subsumed by that. All the studies I've shown were relatively small, 3, 4, 5 centimeters, which is sort of good, but not good enough. And so we are actively working on incorporation of nerves and muscle, but in parallel looking at the other aspects of pneumatic and hydrostatic devices for peristalsis. With respect to how to check for tensile strength, what we do is we do this ex vivo, and we have straightforward biomechanical devices to assess the breaking strength, essentially. Paolo, I don't know if you have a different view on the nerves.
No, absolutely, David. I would share exactly what you said, and that's one of the reasons why we focus on part of the intestine. So one thing that we are getting more and more use is the fact that the organs that we may engineer will look very different to the organ we are used to have in the body. And it's possible that you know, some mucosa that you would implant somewhere would help you overcoming some of the nutrition problem that you have with short bowel. And that's exactly why we saw that's very difficult complete engineering of the old intestine, and we focus on part of that because of the different component. I mean, Tracy, who is a very good friend and a collaborator on the European grant, and Jay Vakandi, I suppose, have shown it, you know, 20 years ago or more that you can put some cells derived from the intestine that used to be called organoid at that time, but contain also the mesenchyma. And you can have some structure that look like the intestine, but actually the difficult bit is to make that structure working in an appropriate way, as you were saying. So I think it's very, very complex, and the way that we do it, and I think you know, I believe I share that with you and the others in the call here, is that if we understand mechanisms, we go faster to patient. I mean, I always say also to the clinical colleagues that come to the lab that actually understanding mechanisms don't make you more far away from patient, but we go faster because we understand what we can and we cannot do. And also the regulatory are much happier to help us translating something that we know and we understand exactly every single mechanism. So that's why the journey, rather than getting more complex, get simplified, and we try to take out components if we can, because that's put us safer and closer to patient.
Mm-hmm. Okay, thank you. There's another question from Jose Mauchek asking about scaffolds. I bet that's for both of you, whoever wants to answer. Could the gut that is resected during surgery for NEC be used as a scaffold for stem cells? And he mentions like the cells are dead, but the scaffold remains. So would that work?
So, I mean, if I can say from the D cell point of view, I wouldn't use that scaffold for a number of reasons. The first is that when you have ischemia or even perforation or necrosis, you free in the tissue a lot of both mechanical and biochemical results of the necrosis that do not really facilitate cell proliferation or cell seeding or anything. And the structure that results in the scaffold, it's not maintained. So I wouldn't suggest using that scaffold for repopulation, but I don't know what David thinks.
I love the question. It's very creative. I agree with Paolo. I think, there's neck and there's neck and there's neck. So we're aggressive with neck because we think that with surgery for neck, because we think that there's two reasons to operate on the belly. One is to remove the dead bowel, which clearly is dead and of no use. And the second is to protect the brain. As the gut is releasing molecules, and we've identified some of them, they cross the blood-brain barrier and they activate the microglia in the brain and they cause cell death. And that's probably, or at least and the cells are not neurons, they're oligodendrocytes. So you get a white matter injury in humans and in mice that we've induced to develop it. And again, Marty Blakely, to refer to his beautiful work in the Annals of Surgery, neurocognitive impairment is higher in those patients that did not have an XLAP but were otherwise stratified, suggesting that belong to the disease. So with that mini diversion, I think the pendulum is going to switch, or maybe this is a society to help me do this. But I think the pendulum is going to switch, so we're operating a little earlier. Now that will come at the risk of having some unnecessary laparotomies, maybe, but a 1 kilo kid can tolerate an XLAP pretty well generally. Maybe release some fluid, maybe put a silo on. But you may be in an instance where you're removing bowel that's a patchy, that isn't completely frankly necrotic, and maybe if you had the right setup, this is the bowel that you have in mind. And then certainly, but otherwise I agree with Paolo. Otherwise, the stoma closure. We have to be very careful when we're closing stomas that we don't take too much bowel, but especially if it's a short gut kid, I will compromise how beautiful the ends are to maintain length, but never compromise function. And so it's a delicate balance that everyone on the panel faces every time they have a stoma reversal on baby with neck. But if you've got a kid that may have a little extra, then maybe you could do that. I don't want to suggest we would operate for purposes other than to help the kid, but you follow me. So the simple answer is probably not, but maybe there's some nuance there.
Yeah,
Thank you very much.
Oh, sorry, Augustus, just to add it on, that there could be a very nice, understanding what happened to the extracellular matrix in this situation. And that will offer a lot of clues for regeneration, as David was mentioning before.
Yeah, I totally agree with your comments, and also with the comment that we maybe need to operate a little earlier. I really hope that your work and the work done by others showing that these babies might not have a perforation, but their brains and their lungs are actually severely affected. They really would push the surgeons not to put drains and possibly to go to the OR a little earlier. There's a nice, interesting question from Moritz Markel. Originally comes from Martin's group in Leipzig, but he's in Winnipeg doing research. And again, as you lined out the importance of TLR4, so I think it's more for Dr. Hackam, and immunological mechanisms in neck, do you think intestinal organoids or the artificial mucosa can be able to fulfill the complex immunological tasks of the intestine and intestinal barrier? So again, we want more. We want not only that the gut works well, but also that it has a proper immunological function. Have you tested your artificial intestine from this regard?
Sure, it's a great question. Thank you. And they're really important because this is, I suppose, the fourth aspect of the epithelium. There's the nerves, there's the blood supply, and then there's the immune system. And so just 2 points. So to your specific, and please say hello to everyone in Winnipeg, and I hope you're having a good time there. It's a great place. So we haven't actually done immunologic tests on our grafts. We've done basic kind of cytokine release, but we haven't done robust assays for, let's say, antimicrobial peptide release from the Paneth cells, so that we've shown by immunohistochemistry that we have Paneth cells. We haven't shown that they're function, as an example. So I would say we have not done that. It's a good idea. The second thing I would say, though, is the role of TLR4 in the epithelium is probably not immunologic. It's developmental and coordination of the balance between proliferation through Wnt and Notch and death through caspase-3 mediated apoptosis. And so TLR4 has a non-immunologic role in the epithelium, but in the myeloid compartment, lymphocytes and macrophages, the TLR4 is TLR4, and it recognizes LPS. And so I think that's how I would answer the question. I think we need an intact immune system, because whilst I showed some work with treatment of the graft with probiotic, I went through it very quickly, and neck bacteria, that's really just in response to the epithelial signaling. It's not an immune response. And if we don't have an intact immune system, we won't have an intact barrier. And then it's why your question's so good. We'll just have bacterial translocation and we'll be back to square one. So this is another area that we need to work on, and that's getting the right. And also, I would say experimentally, all our human work is in immunodeficient mice. So we're going to have to be smarter about that. Just one final thing about the immune system. We showed in a paper in 2016 that the lymphocytes are critical in the pathogenesis versus protection from neck. And so if we take lymphocytes from a mouse with neck and from the mouse bowel with neck and inject it into a naive mouse, the mouse will get neck. So lymphocytes, and these are Th17 lymphocytes, and the human and mouse intestine has the same lymphocyte population. And then Akhil Maheshwari has shown beautifully that macrophages are pivotal in the protection and induction of neck. And so there's no question that these cells are important just in barrier function in the neonates, and therefore probably in artificial intestine.
All right, then there's another question from Richard Wagner, also from my group, but he's in Boston in Patricia Donahoe's lab at the moment. And he's asking, this question is for you both, did either of you look into how stem cells are affected, like in neck, gut mucosal stem cells in patients, whether these change their molecular fate? So he's asking, or is the epigenetic imprinting of the affected parts of the gut, which could in turn inform which molecular pathways might be beneficial to mucosal regeneration if targeted? Very complex question, Richard, but I hope I transported that well. Do you understand what Richard was saying?
It's a great question. Paolo, did you want to?
Yeah, absolutely. Yes, I mean, we did a bit of work looking at organoids derived from a patient with NEC, and I'm aware of some of the work has also been done by Augusto and Agostino in Toronto about this. I mean, the question is that, I mean, from the organoids point of view, you can certainly culture cells from patient with NEC and from this resected segment. And that's going to intuitive because, of course, the intestine can regenerate. And the stem cell counterpart seems to be more resistant and more resilient to ischemic damage and to necrosis. But we haven't looked at that. Maybe David has more knowledge on the molecular path of the stem cells of the intestine in case of NEC. We haven't looked much into that. We are planning to do it, but it's not the data that we have yet.
Thanks. Yeah. So we've shown that we can culture stem cells from NEC. The work on the epigenetics of NEC and how that is maintained in cultured stem cells has been done from Washington University in St. Louis by Misty Good, and she actually has looked at that. She's presented it. I think she's published it. If not, she should be soon. And your question is answered by her, and that is yes, there are epigenetic differences in NEC that she picks up in the tissues. Whether she did it in the organoids or whether she did it in fresh tissue, I think it was actually in the fresh tissue where she did epigenetic arrays, but it's the same basic idea that you're asking. And we've grown those stem cells, and whether they maintain the memory or not, Misty's work might shed a light, but we haven't looked at that specifically. But it's a cool question, right? Because if you knew that, then that may allow you to reprogram it and perhaps even have a much better mucosal surface to it.
This is a question for Dr. Hackam. Apart from IPCs, are there possibilities to enhance the function of a cell or organoid repopulation? So like with gene correction, this is in the context of genetic engineering or overexpression of specific factors.
Yeah, that's a great question. So the answer is yes, and then I'll give you the caution. So we published some years ago. It was a downstream molecule of PUMA, which is P53 upregulated modulated apoptosis, PUMA, which when induced in stem cells limits their proliferation. And so we used an adenovirus at that point to knock it down, and cells proliferated more. There are scaffolds that release inhibitor of apoptosis, and in fact, adding to the matrix apoptotic inhibitors enhances proliferation of the enteroids, and we're developing scaffolds with additional molecules for that very reason. The concern is any growth factor, and essentially they're all growth factors at some level, has a malignant risk. And so for a kid, that's something that we cannot convince a company to support us on. They're worried about cancer for good reason, and so we're not even sure what the FDA would say. But it's a great question and an area of hot investigation.
Martin.
Well, I don't see any other questions in the chat, but may I ask you, Paolo, when you talked about how to grow epithelium, that brought me to the question, like in recurrent TEF, okay, we see these patients once in a while, and what keeps this refistula open is the epithelium there, right? So you you try to, when you want to manage that open surgically, it's a very demanding procedure. So it's always smarter to try endoscopically to destroy the mucosa. You know, there are people who do chemocauterization of that. So from your research, is there any agent that you use to decellularize parts of the body in a very smart way to solve this kind of problems? You know what I mean? So to sort of reverse everything, not build something up, but very smartly targeted, destroy it.
Yeah, that's a very, very good question. I never thought about, Martin. I suppose we are more aggressive here in London, probably because of our predecessor. So on recurrent fistula, we go back on surgery. But yeah, I mean, there are some agent that may work. The problem is that these chemicals are not so safe to use. I mean, there are some enzyme that could be used, but those enzymes don't really work without the detergent, essentially. And detergent, we know very well what they do to the esophagus. So it's a bit difficult to use them in a localized meta. I mean, it's a clever idea we can look at, but I don't have an easy solution to use a detergent in a easy and localized way without causing any damage to the neighbor cells, and that's a real problem there. But you could think about creating a patch of epithelium that you could use to repair some of the fistulae. And that's something that we have been thinking about. Or for example, on EB, so in epidermolysis bullosa, people have shown it for the skin. There was a Nature paper some time ago showing that you can take those cells, genetically engineer them, and then creating patch of skin. But we have a huge problem in the esophagus, and we could try something similar on the esophagus as well. So that the patch of mucosa, I think, may be something that could be useful there.
That's too bad, Paolo. I was hoping you had a magic solution for us. You know, when we have these kids that have recurrences, they're a nightmare.
Yeah. Next time.
Yeah.
Okay,
Augusto. I think we are
Augusto.
well over the hour, and of course, this is showing you how much interest has been. What is really good in a way to see that there's a lot of trainees and young surgeons that are engaging more and more, and this is what really hope that we surgeon scientists don't become like pandas, but there's actually more people that engage and don't do research just to tick the box for their CV. And so thanks a lot for your inspiring talks. This is really a lot of food for thought and very inspiring for the next generations.
Well, thank you also from my side. I was quite amazed this time. We had a lot of discussion, a lot of questions from the trainees, so you really inspired them. And well, thanks for being with us. We will put that on our YouTube channel, and we cut that section out that David didn't want to share, which is totally fine. There's no problem with that, but maybe the rest of the webinar we can share for the future generation.
Of course. Yeah, with pleasure. Thank you.
Yeah. Oh, that's very generous. Okay, and thank you also, Gaia, for setting this up.
Thank you again, like always. We also put in the chat the Insta and the Twitter account. So ask everyone to follow us on every social.
Very good. So next webinar is in November on biliary atresia, laparoscopic versus open. There's also hopefully a lot of good discussion. And yeah, that's from my side. Yeah,
we have Professor Yamataka from Tokyo, and Barbara Wildehaber, who is the chief of pediatric surgery in Geneva, in Switzerland. So it's going to be very interesting. We're going back to clinical. We'll do some basic science for biliary atresia sometime in the future. There's a lot to do there too. Okay, thanks everyone, and really thank you for taking time from your busy schedules. Really appreciate it.
Yeah.
Thanks.
Okay.
Thank you.
Thank you,
everyone.
everybody.
Bye-bye. Ciao.
Bye.
Ciao.